So the dominance that Mendel described is called complete dominance. It means that one allele, when you have a situation where you have a heterozygous individual, one allele shows its full phenotype and the other allele is basically silent. We call that complete dominance. So for example, you really only have, if you have two different alleles, you really only have two different phenotype possibilities. If it's homozygous, for example, for a red allele, then it'll be red. If it's homozygous for a white allele, it'll be white. And then there's really no other option, because if white's the dominant allele, then a heterozygous would still be white. If red's the dominant allele, then a heterozygote would still be red. And so you only have two, there's only two possibilities on this. There's really no range of phenotypes, there's just two. There's a discrete number of phenotypes, two in this case. Incomplete dominance is where the heterozygous individual has some kind of phenotype that's sort of in between the two extremes. So instead of being just white or just red, you might have a heterozygote that is some kind of pink. So another, I guess, special case that Mendel never had to try to figure out is something called codominance. Codominance is it's a very narrow topic. It really only applies to antigens, which are glycoproteins on your cells. And probably the most common example, or the most well-known example of that, are the ABO blood groups. There's lots of other blood groups, but ABO is pretty well known, so it makes a good teaching example. But these types of antigens, these are the only examples I've ever seen used related to the term codominance. And so that's one thing to keep in mind. So codominance means you can have a genotype and there can be two alleles that are considered dominant. And when both of them are present in the diploid individual, you can show both phenotypes at the same time. So, like I said, this is really just blood types and tissue types. So for example, there's an allele, the A allele for blood type, there's a B allele for blood type, and you can have a genotype that has one A allele and one B allele because they're alleles of the same gene. And so if you have an A and a B, then your phenotype is A and B, because these are glycoproteins that are displayed on cell surface. And so having an A, there's plenty of space on the cell surface. So the cell is able to make the A type and the B type, and both of them can be displayed. And so you can be both. So that's called codominance, when you can be two things at the same time. But like I said, this is just in blood and tissue types. I've never seen an example of codominance in any other scenario. So that's a very narrow topic, codominance. It turns out, though, that ABO blood types can also be an example of what we call multiple alleles. And multiple alleles is like almost all genes have multiple alleles. So whereas codominance is a very narrow topic that would only apply to certain blood type genes or tissue type genes, the concept of multiple alleles is virtually universal to any gene. So any individual gene can have more than two alleles, and almost always does. Even to a point of absurdity, I mean, an allele is just a mutation of an original version of the gene. So there has to be some gene that is originally there. How that first gene got there, that's the topic for a different class. But you know, once there is a gene in an organism, then a new allele can arise if that gene mutates. So, which means that any gene, any new mutation of a gene is considered a different allele. Now, some new alleles don't create new phenotypes, but they're still alleles if they're a change in the genetic sequence, the A's, the T's, the C's, and the G's of the gene. So genes have, it's hard to put a number on it, but it would not be unusual for a typical gene, to have 10 different alleles. So don't get stuck thinking that genes only have 2 alleles. Obviously, ABO, those are 3. That's 3 right there. And there's actually more alleles for that locus. A, B, and O are the most common alleles. But there are other alleles. You can look it up. There's other alleles for blood type. So multiple alleles is really a rule of all genes. Most genes, I can hardly think of one that doesn't have, I can't think of one that doesn't have more than 2 alleles. So that's a rule. So sometimes students, because Mendel only dealt with 2 alleles, they think that any gene can only have 2. Now, any one individual can only have 2 alleles in that individual, but when we're talking about how many alleles can exist in the entire population, lots. So each individual only has 2, but more than 2 exist. Penetrance. Penetrance is an interesting phenomenon. Like multiple alleles, this penetrance is very common. When Mendel did his work, he only dealt with things that were completely dominant or completely recessive. He never had to deal with this issue. But something that we observe in human genetics for sure, and other model organisms. And the definition of penetrance is it's the extent, usually given as a percent, to which an individual with a disease genotype actually shows some of the disease phenotype. So let's say you have 42 individuals. They could be from the same family. They have inherited a dominant allele for polydactyly, which is extra finger. But only 38 of them actually have an extra finger. Now, you're not asked to explain how that's possible. Penetrance is literally just an expression of the phenomenon that some of the individuals, don't have the phenotype. But the number you would use is the number of individuals who have the phenotype divided by the number that have the genotype. So in this example, 38 over 42 is a 90% penetrance. So 90% penetrance is a way of saying, well, most of the individuals who have the genotype also have the phenotype. But if you think about it from the other side, it's also saying that not everyone who has the genotype has the phenotype. So that's an actual thing. And so when you're studying genetics, you always have to have in the back of your mind, especially when you're studying human genetics, that just because someone doesn't have a disease isn't a guarantee that they don't have the allele because of this penetrance thing. So that's always a chance that they don't. And you won't be asked to calculate penetrance necessarily. You're just going to be asked to recognize what it is. And you're not going to have to explain why it happens. Nobody really knows. Now related to the concept of penetrance is something called variable expressivity. And listen to this definition and see if you can tell how it's different from penetrance. It's the degree of severity of the phenotype among persons who have the genotype. So there's an assumption here that of the individuals who have the genotype, they also have the phenotype, but they might have the phenotype very severe or they might have the phenotype very mild. And isn't it possible that somebody has a phenotype so mild that they would be classified as not having it? It's possible. Now, for polydactyly, I guess it's not, but for example, behavioral traits are really hard. You might have a psychiatrist that would say this person does not have, schizophrenia, but maybe they just have it very, 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 very mild. So with behavioral things, it's pretty common to call someone normal when you really can't rule out the possibility that they have a very mild form. But for polydactyly and extra finger, I give you this example. Pretend like these individuals are all related. So they've all inherited a dominant allele for polydactyly from the same family tree, and yet some individuals have a fully formed extra finger and some just have a little nub. The ones that have a little nub, that's going to be easy to correct surgically. The ones that have the fully formed extra finger, that gets more complicated surgically. And then you have this one guy over here who has the allele, I pretend like, has the dominant allele but doesn't even have an extra finger at all. So that could be, that's incomplete penetrance here. The rest of these guys are variable expressivity of the allele because they are expressing the phenotype but not to the same extent. Now, I get to this point and I have to make this statement. Incomplete dominance is different than incomplete penetrance. I know they both have the word incomplete, but don't get confused about the two. Incomplete dominance is where one allele doesn't completely dominate over the other alleles such that the heterozygous phenotype is in between, like pink. Incomplete penetrance is where individuals who have the genotype, who have a disease allele, don't have the phenotype. So those are two very different concepts, although the name is similar. Lethal alleles. Lethal alleles, that's just a general term that could be used to, could be applied to any situation where a homozygous situation results in death. I know your book's going to define it as any allele that leads to death, but that's not how that terminology is used in genetics. We say when a homozygous individual is dead, then that's a lethal allele. Lethal when homozygous. So an interesting example of this is that yellow coat color in mice. Yellow is dominant to everything else in mice. It's dominant to brown, it's dominant to black, it's dominant to white. So if you have a heterozygous mouse that's yellow and made them to another heterozygous mouse that's yellow, you would expect to get 3/4 of the offspring would be yellow, but you don't. And that's because the 1/4 of the offspring that should have the big Y genotype die in the womb. They die before birth, so the yellow allele is a homozygous lethal. It's perfectly viable when in its heterozygous form, but in homozygous form it kills the baby. And again, you don't have to know why it does, just that that's the pattern, that's what this term means. In human diseases, it's pretty common for human dominant disorders to be lethal. For the most part, Huntington's disease always exists in a heterozygous status because homozygotes die before birth. So it's not viable in a homozygous situation.